So I will be starting with the title for today's presentation, which is Human Stem Cells and Effective Drug Screening Platform. So my presentation will be of two sections. One would be the review article, which I'm going to present, and the other one will be a part of my PhD study, which I'm going to explain in brief. So this is the review article, which we published in 2016. It was published in Stem Cell Research and Therapy, and up to date we have Google Scholar citation number as 93. So I would take you through the introduction briefly. So to start with, a healthy body is a human universal desire which all of us want to have. So we do different methods or different chores to maintain this healthy body by exercising, by meditation, or by cosmetic procedures, taking drugs and supplements, or simply by having a balanced diet. In our body, there is a group of cells called stem cells which do this for us. So what is a stem cell? So stem cell is characterized by two main characteristics. They can replicate themselves, that is to make a similar version of themselves, and also they can differentiate into many cell types. And depending on their potency or how many types of cells they can make, these stem cells are categorized. So totipotent cells are the ones which can grow into a whole organism, and pluripotent ones are the ones which can differentiate into cells of any germ layer, and multipotent ones are the ones which can differentiate into cells of one germ layer, and oligopotent stem cells or the progenitor cells are the ones which can differentiate into relative lineage cells, and then we have the last ones are the unipotent or the somatic cells in our body. So that is the lineage of potency or the potential of the stem cells. So moving on to the focus of our review, we focused on human mesenchymal stem cells, which is a part of multipotent stem cells, and they are also categorized as adult stem cells. So these can be isolated from a number of sources, tissue sources such as bone marrow, breast milk, dermis, endometrium, and etc. as shown in the picture, and their differentiation capacity is so wide, so they can be differentiated into bone cells, fat cells, cartilage, skin, and many more types of cells. So about the clinical trials using mesenchymal stem cells, in our review we have mentioned in 2015 when we were writing it, we have mentioned there were 493 clinical trials using mesenchymal stem cells in which bone and cartilage diseases were a priority. But in 2022, just to show you an update, so the number of clinical trials have updated or upgraded, and the numbers have increased. The number is 1,088, and their priority now is to treat COVID-19 using human mesenchymal stem cells. And the range of the diseases has also been changed with the time. So what is stem cell therapy? For a beginner, so this is the pipeline of stem cells. So we extract human stem cells and then we culture them in the labs to increase numbers. Then there is an optional step where we differentiate into respective cell types required for therapy, and then we transplant them into the respective organ locations. So the global stem cell therapy market is huge. It is expected to increase by 20% from the time period of 2018 to 2024. So in our study and in our review, our focus is to tap onto this second step of the stem cell therapy pipeline, where we talk about the stem cell stimulants. So we do have current stimulants to stimulate stem cells to increase their numbers or to differentiate their cell types. So they have been, these stem cell stimulants have been initiated with components isolated from human biological sources. Then, because of the ethical controversies and long, tedious downstream processing, the scientists and the clinicians move towards the recombinant and synthetic stimulants. So these are a few cocktails or few examples of these synthetic and recombinant stimulants which are used currently in osteogenic differentiation, cartilage differentiation, and also for proliferation or to increase number of stem cells. So what about the current stem cell stimulants? Why do we look for novel ones? So the current stem cell stimulants have disadvantages or drawbacks, such as they have high prices because of their cutting-edge, expensive equipment usage and standardizing and so on. So, and then toxicity issues are there, and because of the synthetic material, they are sometimes they are unstable when applied, and they have high rejection rates because of the origin, because these recombinant stimulants come from other animal origins, so on. There are different and a plethora of drawbacks in these current stem cell stimulants. So what do we do? So our focus was to summarize few articles or what write this article, and then we found out that there are natural alternatives in the line, so that it would replace these stem cell stimulants, the current ones, but they are in, of course, they are in the research basis. So these natural stimulants, they come from mostly from herbal remedies. So these herbal remedies, the archaeological proof of herbal medicine dates back to more than 5,000 years, even in Sri Lanka. So 75% of the world population use herbs for their basic healthcare needs, as WHO fact sheets mention. And there is a separate branch of Ayurveda called Rasayana for herbal remedies which are used in tissue regeneration and tissue protective effects. So for Ayurveda and traditional medicine, this stem cell concept is not a new thing. So moving on to the contents of our review article. So here I have listed out the articles or the papers which I have mentioned in the review. So screening for osteogenetic effects on the mesenchymal stem cells. Here you can see in this one, so cell platform, all they have used is bone marrow derived mesenchymal stem cells. So they have used different herbal preparations or different extracts from different traditional medicine systems. Here, this one is from Chinese traditional medicine, and this one is from Korean traditional medicine. This root extract is from Iranian traditional medicine. So on, they have there is a global trend of using herbal remedies or their derived compounds to use as stem cell stimulants in osteogenetic for osteogenetic effects. So moving on to screening for anti-adipogenic effects on human mesenchymal stem cells. Here I have listed out 3 of them. So first one is a herbal remedy, which is a whole extract, and the other one is an aqueous extract. Again, it is a whole extract, and the third one is a compound derived from herbal plant. So here the cell platforms they have used are the bone marrow mesenchymal stem cells, and other 2 are for adipose derived stem cells. So they have checked whether there are anti-adipogenic or whether this prevents being stem cell differentiated into a fat cell. So screening for neurogenetic effects on mesenchymal stem cells, again, I have listed what was written on the paper. So we have 3 extracts and 3 types of cell platforms the same cell but derived from different sources: bone marrow, adipose, and Wharton jelly. So all these have been used to differentiate mesenchymal stem cells into a neuronal lineage of cells. Then moving on to angiogenic and proliferative effects of herbal preparations on these mesenchymal stem cells. The list goes on as olive leaf extract, curcumin, there is a kashaya and a Chinese herbal preparation, and leaf extract of carica papaya and viscum album extract. So here again we can see the cell platforms or the sources of cells are very much differ from each other: bone marrow, adipose, Wharton jelly, dental pulp, and placenta. So not only the good effects or the beneficial effects, we have also listed out the adverse effects on mesenchymal stem cells with these herbal preparations. But the cell platforms, all we could found were the gingival derived mesenchymal stem cells, and there were 3 different types of herbal extracts used, and the effects were either there was no significant proliferative rate or there is a reduced cell viability effect. So although we are focusing or directing other people to use herbal preparations by writing or preparing this manuscript, we also added what are the current drawbacks of herbal preparations. So effectiveness could reduce during mass manufacturing processes, and also there are different solvents used in these herbal preparations. So if not purified properly, they may cause adverse effects. And if local administration is required, then it could be invasive, painful procedures for the patients. And clinical applications are challenging due to variability and complexity of bioactive constituents present in herbal formulations. But recently there is a novel field going on with using synergistic activity and additive activities of herbal preparations. They analyze these for synergistic activities. So this drawback is currently coped up with. So, and then proper standardization and in-depth research is indeed required to ensure safe application in therapy. So our conclusions in this article were that, of course, there are beneficial effects of herbal preparations on the stem cells, and they are widely studied, and these can be used to stimulate stem cells. And mechanisms and modes of actions should be also studied in depth. And although there is a bias towards bone marrow human mesenchymal stem cells, there is a trend towards biological waste-derived mesenchymal stem cells in research and therapy. And also, finally, as the preparation methods are simple, the herbal stimulants will be highly cost-effective. And these can be alternatives to the existing ones with cost-effective, highly available, and being non-toxic. So now I would like to move on to the second section of my studies. So this is my PhD thesis titled Proliferative Differentiation and Toxicological Effects of Selected Herbal Preparations on In-House Established Human Mesenchymal and Hematopoietic Stem Cell Lines. So depending on our outcomes, we have categorized our study into 4 main sections. So the first one is proliferative effects of Ficus bengalensis distillate on human mesenchymal stem cells and hematopoietic stem cells. And the other 3 are of same caliber. So the first one I will be explaining in this presentation. So our screening platform was human mesenchymal stem cells and human hematopoietic stem cells. These are biological waste tissues and blood thrown after childbirth. So we went for this option because these commercially available, there are commercially available stem cell lines, but they are exorbitantly pricey, and with a budget of just above 3 million, we thought of using that money to take more reagents and chemicals rather than using it for a cell line. So we prepared it in-house using standardized methods. So we succeeded, so that is a plus point. So with that, we did proliferative effects, and we also checked for genotoxic effects, apoptotic effects, and because we got beneficial effects or positive effects, we chemically characterized the distillate. So preparation of the herbal distillate was done by our collaborator, Dr. Udaya Samaratunga, senior lecturer at the Department of Ayurveda Basic Principles, Gampaha Wickramarach University of Indigenous Medicine. He is also a traditional medicine practitioner of Madadumbara Rambukwale Potave tradition in Kandy. So he uses both these knowledges, but in this preparation of this herbal preparation, he used traditional medicine methods, and fresh leaflets and fresh aerial crop roots were used to prepare this herbal distillate. So Ficus bengalensis, if someone is wondering what is that, so this is the banyan tree, the common name, and we are all familiar because we have one just in front of our department. So establishing human mesenchymal stem cell lines. So as I said before, we collected human umbilical cord from after a cesarean section, and then we transported it sterilely to our department, to our cell culture facility at CRL, and then these human cord tissue was washed, it was sterilized, and then the arteries and veins were removed, and these were chopped into small pieces, the tissue, and then they were kept in a tissue culture flask for a few days with culture medium and under standard cell culture conditions. And this was the result. So if you can see here, that these thread-like structures are the cells which have come out of the tissue on day 10. There were very few. And moving on to the second picture, now you can see that the number of cells or the attached cells have increased, and this is our tissue which is explanted in the tissue culture flask. This was on day 14. Then on day 20, if you can see that all these lines represent cells. So we have a flask full of cells on day 20. So not only to isolate, we have to characterize these mesenchymal stem cells to confirm whether the cells which we isolated are really the stem cells or the mesenchymal stem cells which we need. So for that, we did two types of characterization. First one was the functional differentiation, where these stem cells were turned into or differentiated into fat cells, cartilage cells, and bone cells. So for fat cell differentiation, we used a different chemical cocktail in the laboratory, which was prepared in the laboratory. And if you can see, when we stain these differentiated cells on day 14, all these lipid droplets in these adipocytes were stained with oil red ochre, confirming the fat cell differentiation. Then moving on to the cartilage differentiation, again a different cocktail of chemicals were used for this. So on day 14, we saw cell aggregates, but we needed to confirm whether these are cartilage aggregates or something else. So the standard method was to stain it with Alician blue to stain glycosaminoglycans in the cartilage cells. So we got a positive result with the blue color, confirming the cartilage differentiation. Then moving on to the osteocyte differentiation. So here again on day 14, we did a differential staining method using alizarin reddes. Here what you see is that the calcium deposits which are stained from alizarin reddes, confirming the osteocyte differentiation. The second method was to check the presence of markers, cell markers of human mesenchymal stem cells on these cells which we isolated. So we used a panel for that, a mesenchymal marker panel, and the method we used was flow cytometry. So flow cytometry was done at Cancer Institute, but the analysis was done by us. So here you can see that the minimum required presence of markers is numbers is 3. So CD105, CD73, and CD90 are mesenchymal stem cell markers. So they were present on these cells, and there is also a marker panel with hematopoietic stem cell markers. So you can see that the negative effect of these hematopoietic markers on the histogram. And again, we did an additional testing to check whether these our cell lines which we are going for screening is genetically stable or healthy cell line. So we did conventional karyotyping, and we received or we obtained results of normal human male and normal human female cell lines. So establishment and characterization of mesenchymal stem cell lines. So the cord blood was extracted from the cord which we collected from the arteries and veins. And then we did magnetic bead selection method to select only the CD34 cells, where these magnetic beads carry CD34 markers. So they get attached to the cells in the blood, and then they are extracted using this magnet shown in the slide. So this is this is what we observed after day 7 in culture, after culture initiation. So there were lots of cells proliferation and initiation. So obviously we used chemical stimulants to take out this outcome. A cocktail of very expensive synthetic stimulants. So it costed around 10 lakhs just for 3 stimulants. So that is why we say that is a huge drawback with these stimulants. So for that also we did karyotyping. And to check on the proliferation and cytotoxicity assays, we did MTT assay where a color formation indicates the increased number of stem cells or cells. And these were our results. So here male and we checked on both male and female mesenchymal stem cell proliferation effect. So the male stem cell line had a significant proliferation rate when treated with this herbal extract. Here, as you can see, this 0.8% had even a better effect than our positive control, which is again a synthetic stimulant called fibroblast growth factor. And effect of these distillate on male and female hematopoietic stem cell line proliferation. So here again you can see there is a significant proliferation rate, but it was similar to the positive control which we use. So the positive control was again that cocktail of hematopoietic stimulants, the commercially available stimulants. So our effect of the distillate was similar to the positive control. Then we went for genotoxic assays after treatment of these because we have to ensure the safety of this herbal preparation. So acridine orange ethidium bromide assay was done. In this, in a normal cell, only acridine orange would bind, and then in a apoptotic cell, ethidium bromide also will go and bind to the cell. So in that, there will be a color difference in an apoptotic cell and a normal cell. So a normal cell would look green and an apoptotic cell would look red. So these are the images or the results of that apoptotic study. So the control means the untreated cells. So the males had 3% of apoptotic cells, and female HMSC's apoptotic percentage was 8.5 in the control line. So for the positive control, where we treated our cells with hydrogen peroxide, had 98.6% of apoptotic percentage, and all the female cells were dead after using hydrogen peroxide treated treatment. So test, we used our extract. So we used our concentration of the extract, which worked as the best. So 2.6% was the apoptotic percentage, but the female line had a 39% apoptotic percentage using this distillate. So this here you can see that the female cells are in red, showing that the ethidium bromide had been bound to the cells. Then we used the COMET assay to check the genotoxicity of hematopoietic stem cells, as they didn't have any significant effects on the female cell line. So we chose COMET assay. So here for the positive control, what you can see is that there is a positive control means the cells which we treated with hydrogen peroxide, they had a tail, extended tail, and for the control and the treated cells with our distillate had no tails. So that means there was no genotoxic effects observed. So because we got beneficial or positive results with our distillate, we went for a chemical characterization. So for the GCMS, with the GCMS result, we found out that cinnamaldehyde was accounting for more than 93% of the distillate, and eugenol accounted for around 3%. So we suspect that cinnamaldehyde is playing a major role in this. So right now we are experimenting on using cinnamaldehyde and extracting cinnamaldehyde from this distillate. We are planning for that and to use that extracted cinnamaldehyde on the cells and check whether it is really the cinnamaldehyde or it is the synergistic effect of all these compounds. So this was other than flow cytometry and going for analysis of chromosomes, which needed softwares and special equipment. All this work was done in combinatorial research laboratory under the supervision of Professor Preeti Udagama. So with the results, we reached out to Colombo Science and Technology Cell. So they helped us out immensely and continuously with to file a patent application. So we filed it in 2019 and we received it in 2021. I think it is the first patent for the department as well. So with our outcomes, we strongly support the validity of drug discovery by identifying active ingredients or whole remedies from Sri Lankan traditional herbal medicine using human stem cells as a screening platform. So I would like to acknowledge my supervisor and mentor, Professor Preeti Udagama, and Dr. Samaratunga, who who is our collaborator, who gives us these distillates to use. And then Mr. Praneeth Ratnayakar for his continuous support in experimental work and presentation work. And also my colleagues, the passed out ones and the current colleagues in combinatorial research laboratory. Also director and staff of CEL, academic and non-academic staff of Zoology and Environment Sciences Department, and also the funding agency, National Science Foundation. Thank you very much for listening.
Thank you very much, Dr. Vindya. It was really nice presentation, and we all would like to congratulate you on your patent, and we all are proud of you. So now the paper and the patent, the presentation is open for discussion. So if you have any questions, you can raise them now. Dr. Vindya, can you hear me?
Yes.
I have a very small question. I don't know. This is related to the methodology, the establishment of cell line. Dr. Vindya.
Yeah.
You used different chemicals to induce cell differentiation, and you nicely showed how those cells were differentiated into adipocytes like that. And it was most of them were differentiated in day 14, right, Dr. Vindya?
Day 14 for cartilage and adipocytes, and there were it took around 21 days for osteogenic differentiation.
Okay, so the question is, how did you keep the stem cells intact without making them differentiating, which where you can use them into your experiments? I mean, you just maintain your normal culturing, or did you take any measures to keep them intact without differentiating?
Yes, they actually, if we don't put any chemical constituents for differentiation, they don't differentiate on their own, so they stay as it is in the culture. So
okay,
it is only for the functional characterization purpose we took a minute amount of our isolated cells and then we differentiated them with the chemical stimulants.
Okay, Dr. Vindya, thank you.
You're welcome.
Are there any other questions?
Vindya?
Yes, Dr. Sachin.
Very impressive work, Vindya. Congratulations and all the very best to you and your team. I just have a small question. So now when you look at osteogenic differentiation, you have specifically looked at osteocytes, right?
Yeah.
So is there a special reason that you looked at osteocytes other than looking at the osteoblasts?
No, it is the standard protocol to look at the osteogenic differentiation, either osteoblasts or osteocytes. We looked at osteocytes because this alizarin redness or the calcium formation is happening on the osteocytes. So it would only stain if the osteocyte is differentiated into an osteocyte.
Okay, thank you.
Yeah, would you like to talk about the gene expression studies that you have planned and the chemical characterization? The chemical characterization you showed, but the functional aspects, the genes and so on.
Yes, so we are actually planning to do gene expression studies as well, because as I said, as we have mentioned in our review also, so there should be a mode of action, and these should be explained when we are saying about bioactivity. So with Professor Inoka, we are collaborating for that to do the gene expression studies. So we are hopeful that we will get good results in that, and we could get a better publication out of it, and we could enrich our results with and experiments with that.
Dr. Vindya, another question, what is your suggestion, how can we use these plant extractions, the herbal extractions to treat which kind of diseases where we can treat using this plant extraction?
So actually, if we use stem cells as a screening platform, there is a plethora of diseases where you can use them for muscular degeneration diseases, where you can by seeing that you are stimulating the stem cells, then whether your stem cells are turning into muscle cells, so likewise, or cardiovascular cells. So likewise, there is a plethora of diseases, but
Yes.
then yes, herbal preparations, in terms of herbal preparations, it is solely that in even in traditional medicine and Ayurveda, they have a lot of herbal preparations for this. So what we can do is we can ask, like we can get them to do novel distillations or novel preparations, and then we can go on to test them and then apply them for different types of diseases. So mostly the bone and cartilage diseases, and also anti-obesity, because we can check anti-adipogenic or the anti-fat cell effects on these stem cell lines. So those are the ones, the main ones which we can test.
Yes, yeah, you're correct. Like, because this is a really good platform, Dr. Vindya, like for the younger scientists, the students, they can come up with a lot of projects, and like we are really proud of you, Dr. Vindya, because this is a really amazing work. So are there any other questions?
Madam, are these stem cells derived from a single individual or several individuals? Could the results be different in different cell lines which were raised using stem cells from different individuals? Do the results depend on the genetic composition of the stem cell line? I have zero knowledge on stem cells. Sorry if these questions are done.
No. So the thing is, we have created cell lines. We have established cell lines around for like from 5 individuals from each gender. So these are average results of these from these cell lines. So to exclude that factor that different individuals would have different effects on these herbal preparations. Did I answer his question?
He has said thank you, Vindya.
Okay.
But can I add a word to that, Vindya?
Yes, madam.
What we have to say is that usually it's homologous. You take stem cells from the first patient,
Yes.
and then you grow them in the lab and put them back into the same person.
Yes.
Right. So we don't use right now, of course, I think stem cells are basically homologous. Your own stuff is put back.
Yes, ma'am.
But I suppose later on, if there are stem cell banks and if these are sort of characterized and HLA characterized and so on, I really don't know about that. Would you know about that, Vindya?
Madam, now these mesenchymal stem cells are tend to like not only they put to the same one, so they have clinical trials going on for like putting them to other individuals as well, like for relatives, and whether it works or not. So likewise, they do have research going on, madam.
Right. So it's like after tissue matching, right?
Yes, madam.
Right. Thank you. We can wrap up now, but I would just like to mention a fact or two. Now, there was absolutely no guide for us, right? I mean, there was nobody to ask, or because to how this was done and so on. We went for workshops, and that's how we started this stuff. So hats off to India, because she struggled for 8 long months to establish the first cell line, and thereafter it was easy. But her perseverance, I think, has to be mentioned. And so all congratulations to you, right? And I would also like to, I mean, like, you know, when you do things in this part of the world and you think this is very rocket science, it's not. It's simple stuff, but it's just that we need to sort of break those barriers and start working on things that we can. Now, ideally, I think the next level, whoever should start working on 3D cultures, right? And I don't think that is rocket science either. It's just that you need a little more money, right? So this is not a one-time thing. And I think whoever who is interested, I think they should take, you know, forge ahead from here, because the basics are now established, right? These are not yet published, but will be published soon. So just to, sort of let everybody know that, when you want to do something, and if you are really focused on it, and you think you can do it, and we always in our lab, we say, yes, we can. And that's how things happen, right? So all the best, Vindya, and again, congratulations from me.
Thank you very much, ma'am.
Yes, Vindya, I'm also agreed to what Professor Preethi Udagam is saying, because like, even though science is simple, like in a practical situation, establishing a cell line is like a very big thing with all the sterile condition you have to maintain. So it is a really good work that you have done, and because it is important, because you have started from the scratch. This is not like a commercial cell line, you just started from the scratch. So we really appreciate your work and we congratulate you. And I also like to thank the audience as well, and also for your team who helped you a lot to do this very fascinating work. And I would like to thank audience also for your to join with us and to listen to this presentation. So congratulations again, Dr. Vindya, and we hope more from you. And we hope another presentation from you with another aspect of your research. Thank you very much, Dr. Vindya.
Thank you very much for the opportunity.